Okay, now let us start our lesson today. This is the last chapter. So it is chapter 14.2, Human Genetic Disorders. Now, have you ever heard the expression, it runs in the family? Look at this. People of African and European ancestry are more likely to have wet earwax. Now, if you look at this wet earwax, this one is a dominant. And there is another type which is from Asian and Native American. That's the dry form that is recessive. So these two different races of human, they have also different types of genes. One is dominant and the other one is recessive. Because of what? It's actually because of in the DNA, if you look at the guanine and the adenine in the gene for a membrane transport protein causes this protein to produce dry earwax. So that's for the Asian and Native American because they have this guanine changed to adenine. Okay, now let's move on. There is a direct connection between molecule and trait and between genotype and phenotype. In other words, there is a molecular basis for genetic disorders. Now, look at this. Genome is all the DNA contained in organisms, and there are around 3.2 billion letters. So remember the code in DNA, right? The code in DNA is what? It's either A, T, C, G, right? Adenine, thymine, cytosine, and guanine. Okay, so that's around 3.2 billion letters in a genome. Okay, and genetic disorders result from there are two types. Remember when we talk about mutations occurs in DNA, and the second one is mutation occur in chromosome. So there's a change in the DNA sequence, or the other one is there's a change in the chromosome, which is the gene inside here. So for example, the DNA sequence, cystic fibrosis. Later on, this one is common as a CF. We'll talk about this later on in details. And for the chromosomal, for example, Down syndrome. You know Down syndrome, right? Okay. Now let's continue. So this disorder, caused by individual genes. So it is because of the individual genes. Okay. These changes often affect specific proteins, associated with important cellular functions. The first example is sickle cell disease. What is that sickle cell disease? Now, this is the sickle cell disease. If you look at the picture over here, this is the abnormal. That's what we call as a sickle cell. And this is the normal one. If you look at this blood cell, this is the normal one. Now, how come that it can become something like this? Because of what? Because the hemoglobin inside, it is pulled one to another, and then they will become something like this kind of shape. Okay, later I'll explain in details. Now, what about cystic fibrosis? Cystic fibrosis is a disease in the lung. So usually those who have this kind of cystic fibrosis, they have difficulties in breathing. Okay, due to what? This is due to the CFTR gene. Okay, cystic fibrosis transmembrane regulator gene. We'll talk about this in details. Now, sickle cell disease, it is caused by a defective allele for beta globin. So if you look at here, this is beta globin. Okay, and then one of two polypeptides in hemoglobin, the oxygen carrying protein in red blood cells. So normally, if you look at here, guys, this is the normal blood cells, right? And then remember the function of hemoglobin. Hemoglobin is a protein in the blood, and that's the main one. What's the function of it? To carry an oxygen carrying protein. Okay, and then it makes hemoglobin less soluble. So due to the defective allele over here, due to defective allele for beta globin, this one, what happened? This will become less soluble. Okay, causing what? Hemoglobin molecules stick together when the blood's oxygen level decreases. Then the molecules clump into long fibers, forcing cells into a distinctive sickle shape. Now, the sickle-shaped cells are more rigid than the normal red blood cells. So that's why this kind of shape, yeah, it is very rigid. This is this kind of shape. Okay, and then what happened? This is the dangerous. And they can get stuck in the capillaries. Remember, yeah, there are three types of blood vessels: the artery, and then vein, and then capillaries. Now, capillaries is the small blood vessels, the smallest one. Remember that. Okay. The second one is cystic fibrosis, or CF. Cystic fibrosis is most common among people of European ancestry. Okay, most cases result from the deletion of just 3 bases in the gene for a protein called cystic fibrosis transmembrane conductance regulator. So we call it as CFTR, cystic fibrosis transmembrane conductance regulator. Okay, what happened with that? Look at this. There's only one amino acid is missing. So you look at here, this is the gene of the CFTR. Okay, this is the gene. Over here, this is the gene. It's supposed to be like this order: isoleucine, phenylalanine, glycine, and then valine. But then look at this. The phenylalanine, it's deleted or missing. So what's going to be happen? The results, yeah, due to that one. So the CFTR normally allows chloride ions, or Cl minus, to pass. Okay, so since this one of the function, remember, across the membrane, the chloride ions, then the loss of phenylalanine, this one earlier, remember, okay, causing the protein to fold improperly. So this kind of protein later on will not be produced or will not function properly because of what? Because it's lost phenylalanine. Okay, and then the misfolded protein is then destroyed. So then this one will be destroyed. Then children with CF have serious digestive problems and produce thick, heavy mucus. This is the problem. It will produce thick, heavy mucus. So what's the problem? They will clog their lungs, and then also the breathing passageways. So people with one normal copy of the CF allele are unaffected. So let's say this is the CF, okay? And if the other one have CF, then it's unaffected, but this one is carrier, okay? Why is it? Because it has this, the recessive, okay? So they need 2 copies of the defective allele are needed to produce the disorder. So if the it's supposed to be like this, CF and CF, see? That must be all recessive. Okay, now let us continue. The next one is Huntington's disease. Huntington disease, yeah, if you look at here in the picture, this is the normal brain, and then this is the Huntington's disease. Look at in this part, in the ventricle, yeah, by the way, in our brain, we have this ventricle. And what happened in the Huntington's disease, there will be something that is enlarged. See this one? So the ventricles, both of them are enlarged. Now, because of what? Huntington's disease is caused by a dominant allele for a protein found in brain cells. So this one now it's a dominant. Okay? And then the allele for this disease contains a long string of bases in which the codon CAG. Now, if you see CAG, it will be coding an amino acid which is called glutamine. And due to this one repeats for around more than 40 times, so it will be causing this kind of disease, Huntington disease. What is that? This is actually mental deterioration, and uncontrollable movements. So they have difficulties in the controlling of their movements, and also they have problem with their mental. So usually, the interesting one, usually they do not appear until middle age. Yeah, and then the greater the number of codon repeats, the earlier the disease appears, and the more severe are its symptoms. Now, there's a genetic advantages, though they get the allele or they will become a carrier, for example, but there is a genetic advantages behind that. So, okay, I'll explain it to you now. For example, here in the US or United States, the sickle cell allele is carried by approximately 1% in 12 of African ancestry, and the CF allele is carried by roughly 1% in 25 European ancestry. Now the question is, why are these alleles still around if they can be fatal? It is dangerous, yeah, for those who carry them. So there's a question now. Is it a good gene or a bad gene? Well, the answer is actually, if they are carrier, then this kind of gene will give them an advantage. That's why we can find something like, we call it as genetic advantage. Okay? Now, what is the advantage? For example, malaria. You know malaria, right? Malaria is a mosquito-borne infection, caused by a parasite that lives inside red blood cells. So malaria is one of the dangerous disease in the world. Okay. Interestingly, you know what? If you look at in the map over here, okay, this is the map of Africa, central part of Africa. Malaria happened here. Most African Americans today are descended from populations that originally lived in West Central Africa, so around this side. Okay, and interestingly, individuals with just one copy of sickle cell disease. So, for example, if I draw sickle cell with S, for example, capital S, and then there's the lowercase s, then due this one, the recessive, remember, it will have very high or highly resistant to the parasite. To the parasite of what? Of this malaria. Yes, that this person is a carrier of this sickle cell disease. Yeah, but then this one, if it is only carrier, which is this one is a carrier, right? It's a carrier because there's only one recessive. It has to be both of them if it will causing as a disease, the sickle cell disease. So what's gonna be happen? This will give as the highly resistant to the parasite, giving them a great advantage against malaria. Okay, that's for sickle cell disease. Now, what about another one? The other thing that is very interesting is this, the CF. Remember, cystic fibrosis. There is one also dangerous disease in the world. We call it as a typhoid, yeah, which is caused by bacterium, okay, Salmonella typhosa or Klebsiella, yeah, but yeah, there are a lot of the bacteria. This one is very dangerous, especially in the digestive systems. It will causing a very high fever. Okay, that kind of disease, you know what? Those who have the CF allele, which is the recessive, let's say this, okay, if they have only one, so for example, this is the CF and CF, okay, so it will help blocking the entry of this bacterium. Individual heterozygous for CF would have had an advantage when living in cities with poor sanitation and polluted water. And because they also carried a normal allele, these individuals would not have suffered from cystic fibrosis. So they are not suffering from cystic fibrosis, but they also have an advantage. What is that? They are resistant to this typhoid. Okay, this is the CF allele. So that's an example of the genetic disorder, the sickle cell disease, and then cystic fibrosis. But then in that disorder, there is a benefit. There is an advantage. Okay. Now, what about the other one? That was in the gene. Now, this one is in the chromosome. Okay. So the second one, we are talking about the disorder in chromosome. One of the famous one that we are going to talk right now is a non-disjunction. What is that? Remember, it has to be diploid, right? The chromosome pairing. So for example, like this, okay, it has to be a pair, okay, and then that's what we call as diploid. So the homologous chromosomes, so for example, this is homolog to this. This one failed to separate. Failed. It's supposed to be able to separate, remember, when it is in the, there is the process PMAT, remember, prophase, metaphase, anaphase, and telophase. During the anaphase, remember, this one will be separated. So one will go to that way, and the other one will separate it to the other way. Okay, so due to that things, it is failed to separate. Okay, so the abnormal number of chromosomes find their way into gametes. So for example, look at this. This one and this one is well separated already. This one and this one is well separated already. Now look at this one. Why it is empty? Because it is failed to separate. Is it dangerous? Yeah. Well, let's say this is a sperm. Then this kind of sperm, it carries 2 chromosomes then. It's a diploid instead of a haploid. So later on when it combines with an egg cell, then there is a problem with the sex chromosomes. The other type is this one. In the first meiosis, yeah, by the way, remember, yeah, this is meiosis 1. Still remember that, right? And then the second one is yeah, this is the meiosis 1, okay, and then this is the meiosis 2, okay, this is the process here. Okay, now what about this one? This is happened in the meiosis 1. It is failed. See, there is nothing. Why? Because this one is failed to separate. And because of this one is nothing, so the result will be empty as well. And this one will be separated here. Now, can you imagine if it is an egg cell? Yeah, let's say this is egg cell, and the other side is sperm. If, let's say, this one meet up with this one, yeah, so what's gonna be happen? 4 then. Or if this one meet with this one, so it will become 3 or triploid. Okay, so later on there will be an example I'll tell you. Okay, here, trisomy. One of the chromosomal disorder is this, having 3 copies of chromosome. Okay, chromosome in trisomy in chromosome number 21. Here we call it as a Down syndrome. I think it is common to you. Down syndrome is one of the disease due to non-disjunction. So 1 out of 800 births, yeah, causes mild to severe mental retardation. This is the sign, guys. There will be this kind of types of finger. We call it as clinodactyly. Okay, and if you look at here, guys, in number 21, yeah, remember what we call this? This is karyotype. Okay, this picture is a karyotype. Remember our lesson last week, 2 weeks ago. Yeah. Okay, so look at this. There are 3, right? 1, 2, 3. So this is what we call as trisomy in number 21. Okay, and then what's the problem? It will causing a Down syndrome. Okay, next is Turner syndrome. Turner syndrome is, this one is in the sex chromosome. So if you look at here, yeah, this one, this is in the sex chromosome. So it has to be XX, right? It's supposed to be pairing like this. But then where's the pair? There's nothing. So this is what we call as X0. What is that? This female, this is a female, yeah? This is a female, but then with only one X chromosome. And this female usually, yeah, is sterile, yeah, because sex organs do not develop at puberty because it has only one X chromosome. Now, that's what we call as a Turner syndrome. The other way around is we call it as Klinefelter syndrome. What is that? If you look at here, the Klinefelter syndrome, it has this. Look at this. This is male, but then it has to be XY, right? Remember? But there is an extra over here. Look at this. XY. There's an extra over here. So that's a male, but with an extra X chromosome. So that is a Klinefelter syndrome. So that's, this kind of male will become sterile. Okay, now let's continue. This is the picture of the Turner syndrome. See, look at this. This is the female. It has characteristics of female, but then it is sterile. And the other one is Klinefelter syndrome. This is a male, but then it has the characteristics of a female. Remember, this one is X0, and then this one is XXY. Okay, this one is no X chromosome, less, and this one there's an extra X chromosome. So look at this. There's the characteristics of the female over here. For example, such as what? Breast development, okay, and then the female type pubic hair pattern, okay, and so on and so on. And if you look at here, they have also wide hips and also the testis, yeah, small testicular size. Okay, so if we conclude in that was in the sex chromosome. If we conclude in the autosome, so autosomal dominant traits, yeah, for example, like this. If you look at in this pedigree, yeah, if you look at in this pedigree, okay, the characteristics of autosomal dominant is what? Appear with equal frequency in both sexes, usually between male. Remember, this one is the male, and then this one is the sorry. This one is the male, and this one is the female. Okay, look at this. It has same frequencies between the male and female who are affected by the traits, and it is not likely to skip generations. If you look at here, this is the first generation, and this is the second generation, and then this is the third generation. Look at that. They have it, they have it, they have it, they have it also on the third, if you look at here. So not likely to skip generations, and then both sexes can transmit the trait to the offspring. So it's either the male or the female, and then affected people must have affected parents. So for example, this one, it has to be parents that is affected. This one, okay, Now the other one is recessive. So in the autosomal recessive traits appear with equal frequency in both sexes, unless penetrance differs in males and females. Okay, so it is equal for both sexes, and then appear only when two recessive alleles are inherited. So, for example, small b and small b. Okay, it has to be both recessive. Okay, and then these traits seem to skip generations. If you look at here, there is none. This is the first generation, right? There is none over here, but there is here in the second generation, which is this one, right? And then there's another one also here, but only one here also. So not all generations. And if trait is uncommon, most parents carrying the allele are heterozygous. So for example, either one of these lead, it has it's supposed to be big B and small b then. Okay, and this one also probably. Okay, now next is done. X-linked dominant traits appear in males and females. It's often more females than males, usually for the dominant. So when we talk about X-linked dominant traits, it is mostly in the female, okay, like in this picture. And then each child with an X-linked dominant trait must have an affected parents. Yeah, must have at least one like this. Okay, and then this one is do not skip generations because it's dominant. Same, yeah, like before. Look at there and here. This is the third, this is the first, and this is the second generation. Okay, they will showing all of the almost it's in all generations for the affected for the traits will be shown up show up. Okay, and then affected men pass the trait to all of their daughters. If you look at this one, interesting guys, this is the male, it will passing all of the daughters over here. Okay, but none of their sons. This is the B. And then affected women pass the trait on to about half of their daughters if heterozygous. So for example here, if it is heterozygous, yeah, if it is affected, okay, this one, see only half. Let's say this is 2, only half. Okay, and then the last one is X-linked recessive traits. X-linked traits are more readily inherited by males because males tend to inherit a trait from unaffected females. These traits tend to skip generations. Sons can only inherit the trait from their mothers, not their fathers. And then if you look at in the picture, all daughters will inherit an affected allele from affected fathers. So for this one, if you look at here, yeah, all daughters, yeah, inherit an affected allele from the father. Okay. Then this is the last one for your exercise, everyone. So if you look at here, I don't know, do you see the number over here, guys? Yeah, if you look at the number over there, yeah, this is 3, yeah, 5, 6, 12, 29, 45, 71, 42. Okay, this one, you cannot see that one clearly, but this is the picture. Okay, draw this one. Yeah, this is the number C. 3, and this is supposed to be 5 and 6. Okay, 12. Okay, and this is 29, right? This one is 45. Okay, and this one is 71, and this one is 42. Okay, I'm not a colorblind, but actually, if I tell you um, my history of my family, my dad is actually a colorblind, but I'm not. Remember, this colorblind has to be the recessive, right? So for me, I am a normal, so in my body I have this, the CB, the big one. Yeah. And remember that this one is attached in the X chromosome. So here in the X chromosome. Okay. So guys, if you cannot see these numbers, it means that probably you are colorblind. Now there's an exercise, just practice with this. Red green color blindness is the inability to distinguish the colors red and green. Now, the gene for this trait is located on the X chromosome. The allele for normal color vision is XB, so they just use XB. Okay, this is normal, so I am this. Yeah, so instead using this one, we are using this one, yeah, the capital B. But the color blind is this. So this is my dad. Yeah. Now the question is, a colorblind woman and a man with normal vision plan to have their first child. Now complete a Punnett square for the couple. So you make a Punnett square, okay, and then complete it. A colorblind, remember the question is colorblind women, okay, with a man with normal vision, okay. And then you see how is the possibility of the first child, of their first child. Okay, do it in a Google Docs. Okay, and then submit it in a Google Classroom. Okay,